Okay, welcome children to class 12 online tutorial. Now today we shall talk about the new topic, that is the chromosome theory of inheritance and the other topics as well. But before that, let us quickly revise till now what we have studied, children. So what we have studied about the concept of the dihybrid cross. It also talk about the concept of the law of independent assortment. Now, law of independent assortment, what it has to say, children, that is the one gene is independent of the other gene. That is, if two genes are controlling two other characters, then at the same time, one gene is independent on the other gene, we can say. Dependent assortment. Now today what we shall learn? We shall learn about the chromosomal theory, chromosomal theory of inheritance, whereas we shall also talk about the linkage and the recombination. That is the linkage and recombination. Also we shall talk about how does the gene mapping takes place. Okay, so we'll discuss today. Now, so let us move on. The first topic that we are going to study is known as the chromosomal theory of inheritance. Now, see, as we all know, the Mendel contribution in the field of genetics. Okay, the major we can say principles laid by was that he told that the concept of genes, or we can say the factors, okay, was being a discrete unit in controlling the character of an individual. So in the year 1865, what he did is the work of Mendel was published. But in those days, we can say there was no means of communication like today that we have. So not much of the work was being recognized from the other side, we can say. Okay, now, so in that case, the communication was not easy, but his work we can say in the later part, that is the later years, that is in the 1900, that is the recognition of the Mendel work was done by the later scientists and the other people. Now, he faced many problems. Why? Because first of all, he was a statistician. I told you in the beginning of the chapter. For that reason also. But at the same year, that is in the year 1902, two scientists who put forwarded their theory, that was the Sutton and the Bovary. Sutton and the Bovary, they, what you can say, came up with the factoids, with the chromosomal behavior. And at that time, the invention of the microscope were also being done, children. Now, what was the, what you can say, differences laid out by the Mendel and the Sutton and the Bovary? Let us study, children. That is, we can say, the Mendel and the other one, that is the Sutton and the Bovary. Now, see, first of all, Mendel, what he did that he gave that. See, there are some we can say factors which are present in pairs. That is known as the allele children. Okay, but on the contradictory, Sutton and Bovary said the same thing, children. That is what the chromosomes. that is the chromosomes which have the genes, which have the genes, or we can say factors at that time, are found in pairs, are found in pairs. Now pairs means that is on the homologous chromosome. Now children, can you tell me what do you mean by homologous chromosome, children? Now homologous chromosome are those chromosomes that is they have the same structure of gene, and the structure and the loci of the genes is also the same. That type of chromosome are known as the what, children? The homologous chromosome. Now the other point of factor was that the factors we can say, or the alleles, they separate at the time of at the time of gamete formation. Okay. Now, on the other hand, they told that the chromosome of homologous pair, okay, chromosome of homologous pair separate during gamete formation. Meaning to say, suppose this is the chromosome, right? Like this is the chromosome. So both the chromosome they separate. One goes on this side and the other goes the other way. That is during the meiosis of the mitotic division, we can say. Okay. Now, the third point of differences was, now third point of discussion was that the alleles or factors independent, or we can say they assort independently, assort independently. Now, on the other hand, Sutan they also give the same thing but in the form of chromosome, children. That is the chromosome assort themselves independently. Now, how we can say, let us see, children. Suppose this is the chromosome, has the gene A, and its allele has the, we can say, small a. Now, the other part of the chromosome, that is this part, has the chromosome B, and its allele, we can say, as the chromosome that is this small b. So, A can bind with capital B or can bind with the small b, and small a can bind with the capital B as well as can bind with the small b. So it can depends upon say. So this is all about the chromosomal theory of the inheritance, we can say. Now the basing on the synthesis of idea, that is the okay by Thomas Hunt Morgan, and a sexual reproduction produced. Okay, Morgan worked with the tiny fruit flies, that is the Drosophila melanogaster, we can say, which was found, we can say, okay, okay, that is the Drosophila melanogaster. Okay, that is, we can say, okay, found suitable for the studies. Now, why? Because this species were being found or being selected because they could be grown, okay, easily in the medium, grown easily in lab, we can say. Apart from that, they are the shorter life cycle for that. So for that reason, it is being done. Now, let us move on to the next part, that is known as the, we can say, that is the linkage and the recombination. first of all, if I want to talk about the linkage and the recombination, now linkage means what? The process by which the two genes combine, we can say. That is the phenomena or the process by which the two alleles or genes gets linked or combined. That is known as what? It is known as what should be linkage. Now, so for the other hand, we can say, if we talk about the types of linkage, so linkage can be of two types, that is the complete linkage and the incomplete linkage. Now we are not talking about the incomplete, we'll talk about the complete linkage. But before moving to that, we can say, okay, let us study about the linkage process or the gene mapping. Okay, let us study about the behavior of the chromosome children. Like, suppose this is the gene or a chromosome, we can say. Now we all know that chromosome are found in pairs children. So suppose this is the gene A, which is located over here, or the loci of the gene, we can say. So if this part of the chromosome gets, we can say, linked over here, तो क्या होगा? There are chances, there are 100% chances that the gene will combine or the linkage will happen. Now in the other case, what happens over here is that, suppose this is the chromosome. Now the gene over here is over here at this, the loci of the gene B is over here. Now if this chromosome, that is this chromosome, allele, this part, if it gets linked, तो क्या होगा? There are less chances that the linkage will occur. Okay, as because the loci of the gene is what at the same part of the chromatid of the chromosome, we can say. So in that case, it will not happen. Now, what if we suppose the chromosome, we can say, is over here? So the linkage will be again, the combination will be less children. Okay, and what if the linkage occurs over at this part very close to each other? The linkage will not happen, or the, we can say, crossing over will not happen. Okay, now crossing over means what? The phenomena by which the two chromosome gets crossed over with each other. That is known as the what children? Crossing over. Okay, now so we can come to a conclusion. The distance between the two genes, if it is more, the probability of the gene mapping or the linkage will be high. यानी कि जितने ज्यादा एलील्स जीन्स दूर उतना ज्यादा चांसेस ज्यादा okay crossing over का. Now let us talk about, come back to this point, that is the complete linkage children. Now complete linkage meaning that is the linked gene does not get, we can say, separated during the okay the during the mitosis or the meiotic division, we can say. Now the best example that we can study is about the Morgan Drosophila. Now the Morgan Drosophila, we can say, had the characteristics of two traits. That is, two traits were being studied at a time. Like the dihybrid, we can say. Now, so the dominant gene, what we can say, of the gray color and normal vein, and the recessive part, what being of the, okay, that is the black, and the vestigial we can say vein. Now, so what happened was that the gray color, that is the GW, or we can say GGWW, and for black that is the small g, small g, small w, small w. Now, if we talk about the dihybrid cross, we get the F2 phenotypic ratio is what? 9 is to 3 is to 3 is to 1. But what if these genes do not separate? They get linked. Okay, वो अलग ही नहीं हो रहे at the time of gamete formation. So what will happen is that you will get, we can say, this one, 2 is to 2 is to 2 is to 2 ratio. That will give what? 2 is to 2 is to 2 is to 2 ratio. But now this is not as a part of discussion we are letting over here. So that is the, we can say, the example being given to you. Now the other example can be given of the, we can say, the cotton. That is the cotton plant. Now this cotton plant having the color of the corolla or the corolla. Now what does the farmer do especially? The linked genes, they do not get separate as I told you. So in that case what happens is that farmers know the end product of the, okay, cotton. That will be the color of this cotton only. So in and all, there is one advantages of those. That is, they are used as marker. They are used as what, children? Marker. Marker meaning the genes can be used to mark the end product of the, okay, F2 generations. So in that case, they know that this is going to be color. This is going to be the being the color of the, what we can say, cotton or the plant we can say. So that is being used, children. Okay. Now the next topic, that is known as the, we can say, gene mapping. Now see gene mapping. See gene mapping means the process by which the two genes they get linked. Okay, uh, that is known as the gene mapping, or the position of the genes on the chromosome. That is known as the, what, children? Gene mapping we can say. Now see gene mapping meaning. Let us take an example. That is, there are the two chromosomes. This is the chromosome. Gene A is located over here, and gene B is located over here. So when the crossing over takes place, तो क्या होगा? Maximum chances of crossing over. That is maximum chances of gene mapping. Now this is the first case. Now what about the second case? This is the A chromosome, A genes, and this is the other B genes. These are the chromosome, the allele we can say. Now it gets linked. So the probability will be less compared to that of the, what, the first part. Now let us take up the other example, that is A genes and the B genes. Now what if the linkage occurs? Again the chances will be less compared to that of the second part we can say. But what if the fourth condition takes place? Exactly over here, तो क्या होगा? More is the less chances of the gene mapping as such. Okay, now let us take an example to study over here better. Is that, suppose this is the chromosome, okay, right? Now the location of the two genes are over here, that is the A and the, we can say, B. Now see gene mapping can be studied by, or can be calculated by two methods. One is the recombination frequency and the other one that is by the map units. Now let us study this example, that is this A and B genes on the chromosome, they are, we can say, separated by 10 units. So what will be the 10 units? That is the distance between two genes, that is A and B, is 10 map units apart. 10 map units apart. Meaning, so the recombination frequency is how much? Only 10%. Now how 10%? Let us say this is the gene of all the chromosomes we are located over here, and the distance between these two, one part of the end and the other part is, we can say, 100 units. Now it says that is only 10 units. So how much percentage will be getting? 100 by 10. So it will be 10%. So the recombination frequency is only what? 10% we can say. Okay. Now let us study about the other aspect, that is the, like in say there are three genes over here, A, B and the C gene. That is A, B, C genes allocated. Now it says that A and B are, we can say, 20% apart, or we can say they are having 20% of recombination frequency, whereas the A and C are having 10% recombination frequency. Okay, so on the chromosome, let us draw the map units. So it is saying that A and B are how many apart, children? 20 map units. Okay, that we understood. But what about the AC? अब there are two probabilities. या तो C यहाँ पे हो सकता है, which is having, we can say, 10 units mapping, or we can say it can be over here also. That is, we can say, 10 map units. So I can write two ways. I can write the structural component of the gene or the location of the genes. That is the first case. That is the ACB. ACB. Okay, and the other one, that is the CAB. That is CAB. Okay, so this is how the gene mapping is being done, children. Understood? So this is how the gene mapping can be done, children. I hope you are understanding the topic that I taught you. And the next topic that we are going to study is known as the polygenic inheritance. Okay, now polygenic inheritance, okay, is mean what, children? Okay, now see, if you have seen in the Mendel's contribution, we know that the, okay, color of the flower are discrete. We can say it either, we can say, purple, or we can say white. Okay, but look, if you look around over here, then there are many traits which are not so distinct in the occurrence and are spread across the gradient, we can say. Now we can say, let us take as the tallness, or we can say height of an individual. That is the height of humans. That is, the height of humans can be either tall, short, or we can say medium. There are three types of people on the basis of the height of the humans, we can say. So in that case, the traits which control the trait of a character by three or more genes, they are known as the polygenic traits, we can say. They are known as the, what we can say, polygenic traits. That is, so if we want to define polygenic traits, that is the a condition of a character where the trait is controlled by three or more genes or factors. It is called polygenic traits. Let us say an example, जैसे मैंने आपको बोला अभी. Let us say an example that is the human tallness or the human skin also. Like I can say, assume that there are three genes, A, B, C genes. There are three genes. Okay, these three genes we can say are responsible for, we can say, the dominant form of any character, like we can say skin. So the recessive part will be what we can say A, B or C. So then A can combine with small b, small c. B can combine with small a, small b, small c. C can combine with small a, small b, small c. So in that case, okay, the genotype with all the dominant we can say that is the AA, BB, CC. Okay, we'll have the darkest color, and with all the we can say recessive color that is the AA, BB, CC, we'll have the what we can say the lightest color, or the we can say the white color. But what if in present in between these two? तो वो क्या होगा? It will be present at what type we can say the brown color skin we can say we have. Okay, so that is all about we can say the polygenic inheritance we can say. Okay, now the last topic that we are going to study is about we can say the Leo we can say trophy. Now pleiotropy we can say is a condition wherein a single gene can exhibit multiple expression or can exhibit multiple phenotypic expression. Okay, now over here what it has to say children, that is suppose this is the tallness of a gene which is responsible for the height. Okay, height of an individual, right? But let us say this gene can also express the color of an individual. तो क्या होगा? So in that case, that condition is known as the what children? Pleiotropy we can say. Okay. Now, example of this we can say is the known as the phenyl ketonuria. Now see phenylketonuria which occurs in the humans. This disease, it's a type of disease children, which is being caused by the mutation of a gene that codes for the enzyme phenylalanine hydroxylase. Okay, now this hydroxylase, okay, what does it do? It reduces the we can say the color of an individual and the skin pigmentation also, as well as the reduction in the hair. Okay, so one of the genes, okay, this enzyme expresses one or more characters at the same time. As a result, what happens is that the many symptomatics occurs like the mental retardation and reduction in the hair and the skin pigmentation. Now, normally this occurs in the various other organisms. Also, we shall study in the detail in the next class. So, I hope you are what you can say clear with the topic and the discussion we had so far, Chintan. After seeing this video, if you have any doubt, then please do let me know so that we can solve it. and move on in the other part, that is the sex determination, and the sex determination in the humans and the honeybees, and the mutation in the genetic disorders, we can say.